Hello guys, welcome to my channel Bioslides. Hope you are all doing well. So this is my second video and today I will be presenting on the topic ELISA or enzyme linked immunosorbent assay. So if you are preparing for several competitive exams like CSI or DBT, then this video will be helpful for you a lot as this is an important topic for all those exams. So without wasting much time, let's move on to the video. ELISA is a plate-based assay technique. Basically, you must have done a plate assay. There are several kinds of plates. I will give you the example of a 96 well plate. So this consists of about 96 small wells that are present within the plate. And onto each well, we would be adding the reagents and samples needed to carry out the reaction. And this ELISA technique is used for dictating as well as estimating the amount of small molecules or macromolecules like peptides, proteins, antibodies and so on. And for dictating the presence or to estimate the presence of this substance, we use an enzyme conjugated antibody which would react with a colorless substrate in order to generate a colorless product. And we will be estimating the amount of color that is produced there, and this substrate is often called as a chromogenic substrate. Some of the common examples of the enzymes and the substrate used include alkaline phosphatase, which uses orthophenyldiamine dihydrochloride as substrate, or horseradish peroxidase, which often uses para-nitrophenyl phosphate as its substrate. In case of ELISA, we are first coating a polystyrene well plate with either the antibody or an antigen. So if we are coating the well with an antibody, then in this case we will be introducing the serum or the sample containing the antigens, which will be probably binding to the antibody. The inverse is also possible, that is, if you are coating the well with an antigen, then we will be introducing a sample containing all the antibodies that would probably interact with the coated antigens. And after incubating for some time, we will be using a wash buffer to remove any unbound antibodies or antigens present within the well. So if you consist of a mixture of antibodies and you are suppose that you are introducing this mixture of antibodies into an antigen coated well, in this case some of the antibodies will bind to the antigen while others will remain unbound. So when we are using this wash buffer, we are probably removing these unbound antibodies from the well so that only the antigen antibody interacting complex will be present within the well. And after that we will be introducing secondary antibodies that will consist of an enzyme attached to it, like I have given a previous example of peroxidase or horseradish peroxidase alkaline phosphatase. So this antibody or secondary antibody will have affinity towards the SC region of the primary antibody that we are adding to it, or the antigen or the epitope of the antigen that we are earlier added to the well. So after incubating for some time, the unbound antibodies will be washed out. Because even if you are using secondary antibodies, there will be excess of it which might not bind to the antibody or the antigen respectively, and this has to be washed off. So in this case we are also using the wash buffer again for this process. And after incubation for some time, we will be adding a suitable substrate into it. So the substrate will react with the enzyme if the secondary antibody will be present within the well. Even after washing, the substrate will react with it to produce a color. If a secondary antibody will not be present in it, then this substrate will not react with it and we will never get a color. So if we are getting a color, we can measure the intensity of color by measuring the optical density at 450 nanometer, and the intensity of the color will also give an estimate of how much concentration of that particular antigen or the antibody will be present within the sample. Basically, ELISA is of four types, which is dependent on the base of binding between the antigen and the antibody, and these are probably direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So, what is direct ELISA? In this figure, you can see that there's an antigen coated well. So the antigen are represented as small blue circles which are coated to the surface of the well. And after that we are washing the well so that to remove any kind of unbound antigen that is present within the well. And after that we are adding the enzyme conjugated antibody to the well. So if the enzyme conjugated antibody has any specificity to the antigen, it will bind to the epitope that is present on the surface of antigen and remains bound to it. And this enables the formation of an antigen antibody complex. So in this case, after when even if we are washing out the well, the antibody will remain bound to the antigen. And after that we are introducing the specific substrate that would react with the enzyme and which would probably give the specific color. So the next is the indirect ELISA. So the only difference between an indirect as well as direct ELISA is that in case of indirect ELISA, we are using two different kinds of antibody. One is a primary antibody and second is a secondary antibody. So the primary antibody would bind to the antigen, while the secondary antibody would be binding to the constant region or the Fc region which is present within the primary antibody. And in this case also we will start with the antigen coated well, and we will be giving an appropriate wash in order to remove the unbound antigen that is present within the well. And after that we will be adding the primary antibody. So the primary antibody will be binding to the epitope that is present on the surface of the antigen, and again we are giving a wash to remove any unbound primary antibody. And after that we will be adding the secondary antibody, which is often linked to an enzyme, and this will in turn bind to the Fc region or the constant region which is found within the primary antibody. After that we are again giving a wash to remove the unbound secondary antibodies, and then we are introducing or adding the substrate which would often react with the enzyme to give a particular color. So this is the basic difference. We are only introducing a specific type of two types of antibody in case of indirect ELISA as compared to only one type of antibody which is found in direct ELISA. The next one is the sandwich ELISA. So this will be similar to an indirect ELISA, but in this case we are coating the well with an antibody rather than the antigen. And here we are not considering the antibodies as secondary or primary antibodies. However, we are using the same antibody in alternate series of introduction uh, through each step that will be carrying out within the sandwich ELISA. So in this case you can see that first we introduce an antibody coated well, and then we are washing it to remove any kind of unbounded antibodies. After that we are adding the sample containing the antigen. So the antigen would probably bind to the antibodies or the antibody which is coated within the well if it has specificity to it, and after that we are washing it out in order to remove any kind of unbounded antigen. And after that we are adding the same antibody, however conjugated to an enzyme. So in this case we are using the same antibody. There is no distinction like primary or secondary antibody. Here we are using the same antibody having the same specificity to the antigen. However, the second antibody we are adding should have an enzyme linked to one of its ends. And after that we are washing to remove any unwanted interaction or any unwanted or unbound antibody complex. And then after that we are adding the substrate to it, which would probably react with the enzyme to give out a specific color so that it could be later be measured. So the last type is also called as competitive ELISA. So in this case, it is totally different from the first three types of ELISA that we have previously discussed. So in this case, first we are incubating the antibody with the antigen that is to be measured. So this process is actually happening because we want to increase the sensitivity in this type of ELISA. And after that, we are introducing this specific complex, that is an antibody antigen complex, into the antigen coated well. So in this process, if the antibody has more specificity to the antigen that is present within the well, then a process called as competitive binding actually happens. So the antibody will leave the antigen that is already bound to and then binds to the antigen that is present within the well. And after that we are washing out to remove any kind of unbound antibodies, and after that we are introducing the enzyme conjugated secondary antibody to it, which will bind to the Fc region that is present within the primary antibody, followed up by introduction or addition of a substrate which would probably react with the enzyme to give a specific color. So there are several applications that come under ELISA. Some of the common types are given below. First one is the determination of concentration of antigen and antibody which is present within the serum sample. It has already been discussed previously. And next one is the early detection of cancer. There are several types of markers that distinguish a specific type of cancer, right? So if you have specific antibody that could detect this specific type of marker, then this ELISA could be used to detect the presence of cancer in an individual. And also in case of pregnancy testing, in case of urine, there is a hormone called human chorionic gonadotropin which is a prerequisite for pregnancy. So the presence of this specific type of hormone can be used to confirm the presence of any pregnancy in that individual. Also to test the presence of any drug like opiates, cocaine in urine, and also to determine the concentration of food allergens even in small scales like parts per million scale can be detected using ELISA. And last one is the detection of virus. And this ELISA was the first gold standard test to detect the presence of HIV within an individual. So this basically measures the amount of a particular protein called as cystatin C within blood sample. So higher the amount of this protein, higher will be the level of HIV infected cell that will be present within the blood sample. So that's it. I hope that you all like the video, and if you have found the video to be quite informative, do hit the like button and also subscribe to my channel as I will be uploading new videos whenever possible. And hope you all have a nice day, and thank you.